So welcome everyone. It's my pleasure to host the CHAPPEL lecture this year. My name is Dr. Lauren Grant and I'm an assistant professor in the Department of Population Medicine. Before we begin, I'm just going to read a land acknowledgement. So we acknowledge that the University of Guelph resides on the ancestral lands of the Attawandaron people and the treaty lands and territory of the Mississaugas of the Credit. We recognize the significance of the dish with one spoon covenant to this land and offer our respect to our Anishinaabe, Haudenosaunee, and Métis neighbors as we strive to strengthen our relationships with them. Today, this gathering place is home to many First Nations, Métis, and Inuit peoples, and acknowledging them reminds us of our important connection to this land where we learn and work. Okay, so I'll invite you to settle in because it's going to take me about 5 minutes to get through all of the accolades of Dr. Brian Coombes, and that's if I talk really fast. So thanks so much for being here today for OBC's CHAPPEL Memorial Lecture Series. Held annually since 1988, the CHAPPEL Memorial Lecture Series has hosted numerous internationally known scientists who have inspired and motivated our graduate students, faculty, and staff to achieve the highest possible goals in basic or applied research and to inspire our undergraduate students to consider alternate career paths and become leaders in their profession. This lecture series was established in 1987 through an endowment from Ontario Veterinary College graduate, Dr. Clifford Chappell, who I understand is online today, and his wife in memory of his father, Herbert James Chappell. Thank you so much, Dr. Chappell. I'm pleased to introduce our speaker for this year, Dr. Brian Coombes, professor and chair from the Department of Biochemistry and Biomedical Sciences at McMaster University. Dr. Coombes is an internationally recognized expert in infectious disease and host microbe interactions, focusing on gut health, chronic inflammatory disorders, and antibiotic drug discovery. His research combines molecular microbiology and preclinical models to uncover how bacterial pathogens influence disease and immunity. His lab collaborates widely with industry partners on novel therapeutics for Crohn's disease and antibiotic resistance. Dr. Coombes is a fellow of the American Academy of Microbiology and recipient of numerous awards, including Canada's Top 40 Under 40, the Public Health Agency of Canada's Scientific Merit Award, and the Fisher Scientific Award for Career Achievement. He has published over 220 papers and abstracts and delivered more than 80 invited talks worldwide. And here's where I'm going to add in a few more details. So I was very fortunate to be amongst Dr. Coombes' first crop of students, first as a summer student, then as an undergrad thesis student, and then as a master's student. And that allowed me to witness both his brilliance, his creativity, his work ethic, and his steady leadership that underpins all of the achievements that I just mentioned and continues to inspire me and many others. So without further ado, please join me in welcoming Dr. Coombes.
Thanks very much, Lauren, for that very kind and generous introduction. I feel like the Top 40 Under 40 award is starting to get a little bit cheeky because it's so far in my rearview mirror now because I'm well past 40. But thank you for sharing that. Okay, so today I'm going to talk to you a little bit about what my lab is interested in the realm of antibiotic drug discovery. We focus a lot on zoonotic pathogens. I'm going to be talking mainly about Salmonella today. My lab also works on many other types of pathogens that are involved in other kinds of bloodstream infections. We also work on microbes involved in Crohn's disease, and those are also interesting stories, but I don't have time to talk to you about them today. I will be very happy to share some vignettes about how we got into this work, and then the last half of the talk I'll share some unpublished stuff that I've actually not published and not presented before, so hopefully that's interesting to you. I was also asked, because there's a lot of students in the audience, I was asked to sort of share a little brief bio or pedigree of how I got to where I am. And so I started at McMaster University, and in fact, before my PhD, I did my undergraduate degree there in biochemistry. I stayed on for my PhD at McMaster, and there I did a PhD in medical sciences. I worked on sexually transmitted infections, including chlamydia trachomatis and chlamydia pneumoniae, which is an airborne pathogen. And in grad school, I learned a lot of interesting things, and obviously things that are unrelated to science as well. I learned how to think. I learned how to be creative and ask important questions. I learned how to solve problems, how to choose a good question that's going to obviously get you papers, because that's what we're trying to do as grad students, is publish. And I learned that and I didn't actually know this going into grad school, but I loved working with my hands. I loved running around the lab. I loved being on my feet. I loved pipetting. And it's funny, I don't do that anymore, but that's where it all started. I loved working with my hands, and I thought this is something that maybe I should keep going with. I then moved to the University of British Columbia and trained with a very well-recognized and famous microbiologist called Brett Finley. I was in the Michael Smith laboratories on the UBC campus from 2002 to 2006. There I learned a completely different set of skills. So there I learned how to write. I was writing papers more often. I was writing grants to help fund my postdoctoral fellowship. I learned how to be independent, because at that point as a postdoc, you're trying to learn how to operate as an independent person. I learned how to build my networks, to go to conferences and overcome the fear of going up to the big wigs in the field and speaking to them and telling them who I am. I learned how to collaborate for value, so getting into collaborations that I know will enhance my own research, but also enhance the work of others. And how to choose a fundable problem. There's lots of interesting things to study, but you really got to work on something that's going to get you a grant. And so that's one of the other interesting things I learned. And I learned how to be a mentor to others in the lab and to be mentored myself. I had the great fortune to be recruited back to McMaster in 2006 to start my own independent lab as an assistant professor. I also when I was first recruited to McMaster in 2006, I was part of this really interesting program with the Public Health Agency of Canada called the Interchange Program. And so in addition to my role as a faculty member at McMaster, I was also appointed as a research scientist with the Public Health Agency of Canada in the Laboratory for Foodborne Zoonosis, which I'm sure many of you based in Guelph will know very well. And that was a very, very interesting and important collaborative relationship when I first started my group. As a faculty member, I have learned many, many things, and I still continue to learn, but most importantly, I learned how to lead and how to listen, importantly, how to raise money, how to guide, and that PI actually stands for perpetual improviser. Because now, when you first start your lab, you do so many things that you were never really trained to do. Underpinning it all, though, is the science, and so that's what I'm going to start to talk to you about now as well. That little thing is there. I don't know if we can get rid of that, but okay. So this is, I'm going to just sort of share when I first started my group, these are the kinds of problems that we were trying to think about. And this is all leading us towards antibiotic drug discovery to find new drug targets to combat antimicrobial resistance. So that's the framework. We were less interested in gene content as new targets, and we were more interested in how genes are regulated. And so we got very interested in this idea of what we call regulatory evolution. And this is really just heritable genetic change in how genes in certain organisms are wired and fired in different environments. So to just walk you through this, so imagine this could be a bacteria, let's say. It's got a gene that's regulated by this cis regulatory element upstream of it, and this element is recognized by this particular orange regulator. And so when this transcription factor binds to this regulatory element, it can induce the transcription of the downstream gene. But of course, bacteria are promiscuous. They can grab DNA from the environment and plasmids and other types of horizontal gene transfer events. And once in a while, through those events, they pick up other regulatory factors, transcription factors, which are at first they're foreign to the bacteria. The bacteria doesn't really know what to do with it. So they find themselves with this new regulator with nothing to regulate. And over time, through Darwinian selection, these upstream cis regulatory elements can evolve so that over time, this regulator can now recognize a DNA binding sequence upstream of this gene. And now the bacteria has it can make a choice. It can either maintain this original regulatory connection, so this gene is continuing to regulate it by this orange regulator, or it can choose to now regulate this gene with this new red regulator. It can work almost like a Boolean logic gate, so it can do both of these things. It doesn't have to get rid of one regulator, it can keep both. And these regulators are then controlled, and they control gene expression in different times and places. And so maybe when bacteria are living in the environment, this regulator is dominant, but when they're infecting a host, perhaps this red regulator is dominant. And what we really got interested in as we started the lab is trying to understand in Salmonella specifically what genes in this bacteria have undergone regulatory evolution and what genes have been subject to this kind of regulatory co-option so that we can better understand how regulators that are only active during infection, how are they regulating these genes and what are these gene products? And our thinking there was that if we could find out the genes that have undergone regulatory evolution to only be expressed in the host, that might direct us and guide us to new drug targets. And so this was what we started with. So regulatory evolution controls bacterial phenotypes, including virulence and antibiotic resistance, and that was the framework. These are the motley cast of crew who joined on the Intrepid journey. These are the Coombes lab OGs. Suzanne Osborne was my very first PhD student, and Anna was also another very early on, or Anna was my first PhD student. Suzanne came along the next year. Colin Cooper, David Mulder did some amazing bioinformatics work with us. You'll know this character here. I knew her as Lauren Waller at the time, but now she's Lauren Grant. She hasn't changed a bit, I don't think. She was a master's student in the group from 2010 to 2012. Brian Tunema also did some amazing stuff in Bushra, and this was the crew really that were tackling some of these problems. And so how did we do this? We set up the basic scheme, and that is we all know that an organism's genotype drives its phenotype, and so this is if a bacteria has a certain genotype, the genes are expressed under certain conditions, and they give rise to this bacterial cell, and this cell can metabolize and divide and do all the things it needs to do. But again, because bacteria are promiscuous, their genotype is not static. They can evolve by mutations, they can evolve by taking up plasmids, they can get infected by a phage and gain a whole bunch of new genetic information. They can be co-opted by transposons, which also bring genetic information. And so the genome is constantly changing. And as bacteria evolve through these types of events, their phenotype can change. And so although the core genome stays static, for the most part, these accessory genes can then change what the bacteria does. And it can change its virulence properties, it can change its antibiotic resistance, it can change where it's able to grow and how it's able to defend itself. And then there's this overlying third regulatory level, which is this idea of regulatory evolution. And that is with this genotype, the bacteria can evolve how the genes are regulated by changing those upstream coding regions, the non-coding DNA. So the gene content doesn't change, but the non-coding DNA or these regulatory elements do change. And now the cell, with different types of regulators and different types of binding sites for transcription factors, the cell can make interesting decisions, like where it is, who is trying to kill us, are they our neighbors, what are they trying to do, what do they want me to do, and what resources are available to us. And so again, with a single genotype and overlying regulatory evolution, a single bacterial cell can adopt multiple types of phenotypes. And it's the phenotype of growing in a host that we're most interested in. So we got interested in questions like, does regulatory evolution, or how does regulatory evolution help us understand things like virulence, the ability to infect, cause disease, evade natural immunity, evade antibiotics, for example? And can we exploit this knowledge for new antimicrobial drug discovery? Because even back then, antibiotic resistance was a problem, and now it's an even greater problem. So the answer to this question we thought was yes, because what we did learn from the early group of folks I showed you through the several papers was that the genes that are most subject to regulatory evolution happen to be genes which are essential during an infection. And so you could change the way the genes are expressed, put those mutants back into a mouse, and those mutants, which are only mutated in regulatory sequences, were highly attenuated or in some cases completely avirulent. And so that told us that the regulatory sequences were actually under selection. And that these gene products were important only in the context of a host setting. So you could make mutations in the genes and those bacteria would grow just fine in a test tube. But when you put them back into an animal, they have problems. So what's this concept of in vivo essential genes? Because that's really what we were trying to unlock, is this cryptic collection of genes which have really been untapped in antibiotic drug discovery. So imagine this circle contains all of the genetic information in your favorite pathogen. For us, we're talking about Salmonella today. Salmonella has about 5,000 genes. About maybe close to 3,500 or 3,700 of those genes are what we call core genome, which are common amongst mostly all Salmonella, E. coli, for example, and then a whole bunch of other genes which are accessory genes, which are kind of unique to Salmonella or certain species. The genes that are required to grow in, let's say, LB, a nutrient-rich media in the lab, is only about 300 genes. So that you could whittle this bacteria down and make mutants, and you could make a bacteria with only 300 genes, and it would still grow. So there's 300 genes which are essential to grow in rich media. You can't make mutants in these genes, of course, because they're essential. However, when you shift growth onto a nutrient-limited media, like a minimal media, for example, about 250 additional genes become essential. So these 250 additional genes, you can make deletions of them and they grow on rich media just fine. But when you challenge the bacteria then on a nutrient-limited media, they don't grow. So we call these genes conditionally essential because they are kind of essential, but only when the bacteria is compromised for nutrient acquisition. So these would be genes required for building or uptaking different types of nutrients, pumps, transporters, etc. But the real money is here, which are the genes required to iterate a successful infection in a host. And there, the genes essential to do that is much greater than 550. It's something greater than 550. And for most bacteria, we don't know what that number is. But this you can immediately see is a much larger target gene pool to go after if you're looking at new targets for drug discovery. They're essential in vivo, and this is where we'd be treating infections anyway. And so these genes really get overlooked completely in sort of classical antibiotic drug discovery, where you're growing a bacteria on a rich media, you're exposing it to different small molecules and asking, does it die? None of these mutations in any of these genes wouldn't die, or if you hit one of these genes, it can still grow in LB. But it's the compromised growth in the context of a host that we were most interested in. And so we then shifted our focus to look at high throughput screening under what we call infection relevant conditions to focus our attention on this larger pool of bacterial targets. And so what are some examples of in vivo essential genes for Salmonella? Most of them have to do, not surprisingly, with the innate immune system. The innate immune system is really good at killing things that don't belong. The innate immune system throws all kinds of noxious chemistry at bacteria. And bacteria, if they're to survive that innate immune system, have to find ways to defend themselves against that chemistry. And so in Salmonella, what we've learned over the years is that many of the in vivo essential genes produce gene products required to evade the body's innate immune system. These are just some examples. So imagine this is a Salmonella bacteria which grows intracellularly in many different types of cells, including immune cells, for example, which is a super clever way to live in a body if you're trying to infect a host, if you're a bacterium. Live inside the immune cell that's trying to kill you. And the immune cell does a lot of different things. So it has transporters which pump out divalent metals from the vacuole that the bacteria is trying to grow in. Divalent metals are essential for bacteria, and if they don't have them, they can't divide and grow. And so they try to limit metal acquisition by the bacteria. The immune system will pump in protons to acidify this vacuole and make it really quite acidic, actually, and try and kill things that it's ingested that way. It'll also produce reactive oxygen species to try and damage the membranes of living things that in this case, a macrophage, for example, has ingested. And they can make nitric oxide, which is another type of way that they can damage membranes and other proteins. And they can produce antimicrobial peptides, which are our body's natural antibiotics, for example, which are really quite potent at killing bacterial cells. But in the case of Salmonella, they've evolved essentially sophisticated ways to evade all of these innate defense systems. And so these are some papers which sort of describe some of this work. But for every innate immune chemistry that I've depicted here, Salmonella has evolved sophisticated mechanisms to either disarm, diffuse, detoxify, or in some cases, fortify the outer membrane of the bacterial cell to prevent binding of things like antimicrobial peptides. And so we thought these types of products would be good to go after as antibiotic drug targets, because if we can identify what the gene products are that are involved in this evasion, and we inhibited them, now we can sensitize those bacterial cells to the power of the innate immune system again. And so again, these genes required to evade host defenses are dispensable in vitro. You would never discover them in a conventional antibiotic screening campaign, but they're essential in a host setting. We can't really do drug screening, let's say, in a mouse, and so we have to find ways to mimic the host setting in the lab. Okay. That was the work of two very talented grad students, Carissa Sai, who was a PhD student, and Jordan Perry, who was a master's student in the lab. And this was our foray into antivirulence drug discovery in Salmonella. And so Carissa devised a very clever screen. And so what she did was she came up with a media. In fact, it was a media that I had sort of tinkered around with in my postdoc, and this was co-opted for this screen. But we call this, in the lab, we call it something totally different, but it won't mean anything to you. But essentially what it is a host mimicking media. It mimics that environment that Salmonella is growing in a vacuole in a macrophage pretty closely. And when you do that, it expresses all the same genes that it would if it's like it's growing in a macrophage. And so Carissa developed that media into and miniaturized it into a 3D 4-well plate format. She took Salmonella and made them luminescent, and so they glow luminescent only when they are exposed to this media. So the bacteria thinks they're inside a macrophage. It starts expressing all the genes required to evade the innate immune chemistry, and when it does that, it starts to glow. And then she screened those glowing Salmonella against a small molecule library of about 50,000 molecules made up of various sources in a very small well setup, 384 well plates, 30 microliter cultures, this really cool system we have at McMaster for acoustic droplet dispensing, so we can introduce nanoliter volumes of small molecules into the wells. We let those reactions go for a while, and then over time, whatever time we want to wait, we then put the plates in a luminometer and look for wells which are either still glowing, in which case the bacteria are growing just happily, or look for wells that are dark. And so those would be wells in which the small molecule has inhibited the expression of this particular virulence regulator. This is the primary screening data. Again, so this is luminescence. This is the replica plots. So on the y-axis, we've got one replicate. On the x-axis, we've got the second replicate. So there's good concordance in the screen. Of course, we were looking for things wells which were dark because the Salmonella had either been killed or no longer expressing the virulence regulator that we know is involved in immune evasion. Those would be these guys down here. So these in both screens, these would be low luminescent producing wells. We counter-selected all of this screening data against growth, and so in addition to measuring luminescence, we measured OD. We were not interested in things which were killing the bacteria directly. So we weren't interested in direct acting antimicrobials. We were interested in these cryptic in vivo essential drug targets. And so we counter-selected on growth, and so this is just a replica plot, but instead of looking at luminescence, we're looking at OD. These would be wells which we would eliminate because they are killing Salmonella by themselves. So these would be, again, probably known antibiotics that are in the screening library, in the compound collection, which we were not interested in. So we selected only things which were inhibiting the expression of these in vivo essential genes. Make a long story short, we had another series of prioritization steps. We eventually got down to one, through potency analysis, we got down to one molecule, which we called divastatin, which seemed to be very good at inhibiting the expression of a whole panel of in vivo essential genes. We treated cells with divastatin in a separate experiment and did RNA sequencing and looked at what genes went up and down. And it was very striking when we did this experiment. So these genes so this LPM is this host mimicking media that I told you about. All the genes that are involved in intracellular survival, evasion of innate immune chemistry were all down-regulated when cells were treated with divastatin. And then it also what was downregulated were a whole series of genes involved in fortification of the outer membrane that I told you about before, which bacteria sometimes do to withstand the innate immune system. And so how are these things working? We know that these genes here, which are required for intracellular survival, are regulated by this two-component regulatory system here, this SSRA. This is a classic two-component system in bacteria that probably most people are familiar with, made up of a sensor kinase, which senses some kind of signal in the periplasm, relays a signal through phosphorylation to what we call a response regulator or a transcription factor, and that transcription factor then activates a collection of genes required to survive in the environment and whatever signal that it's sensed. And in this case, these genes are required for intracellular survival, virulence, immune evasion. So that the genes in this column here are all regulated by this guy. The genes in this column are all regulated by these other two-component systems, the FOQ system and the PMRB system, which are involved in many different kinds of adaptations for bacteria, but one of the prominent ones is LPS modifications. And so they make these products of this system make LPS modifications which fortify the outer membrane and make it more impermeable to things like antimicrobial peptides. Okay, so we had this molecule which we think was inhibiting these systems, and so the basic question was, if difastatin is inhibiting these two-component systems, in this case SSRA, we should be able to come up with an experiment where intracellular survival and virulence is compromised in the presence of difastatin. And so that's what Caressa did. She infected bone marrow-derived macrophages with Salmonella, and she either didn't treat them or treated them with increasing concentrations of difastatin. And lo and behold, with increasing concentrations of difastatin, it could inhibit the growth of Salmonella in macrophages. So that made sense with what we know about this system. The second system, these LPS modification systems, we thought, well, if difastatin is also inhibiting these systems, in the presence of difastatin, the bacteria should have trouble making these LPS modifications, and the cell should be more susceptible to things like antimicrobial peptides and some antibiotics, which we know work at the cell wall, like polymyxins. And so we did this experiment where we treated cells, Salmonella cells, with increasing concentrations of difastatin. These are called checkerboard assays. And so this is to look at drug-drug interactions. And so you set up a matrix of one small molecule and another small molecule in increasing concentrations on both sides. And so these are just heat maps which depict growth curves. And so everything in white is no growth, so the cells are dead. Anything in red, the cells are alive and growing. And you can just do this in a plate reader. And so increasing concentrations of difastatin in combination with increasing concentrations of things like colistin and polymyxin B. And what you can see is that difastatin on its own has no growth inhibitory effect. And we expected that because it's only supposed to inhibit growth in the context of a host setting. And so that made sense. But if it was present, then it should be inhibiting these pathways required to evade and resist things like colistin and polymyxin B. And so these checkerboard plots depict what we call synergy, which is as you titrate in increasing concentrations of difastatin, you can lower the concentration of colistin or polymyxin required to kill the bacterial cell. And so, for example, for colistin, for example, it goes from 32 micrograms per mil inhibition down to 1 microgram per mil as you dial in combination with difastatin. So the idea is the small molecule is inhibiting the pathways required to resist these things. And by inhibiting it, we're resensitizing the cell to these antibiotics. Same thing for polymyxin. And interestingly, we saw the same thing for a natural host defense peptide, cramp, which is a cathelicidin antibiotic found in the mouse. So this was all very cool. And we went on to show that this actually works in a mouse. So these are mouse infections where we infect mice with Salmonella. These mice die within about 4 days of an untreated Salmonella infection. Difastatin on its own had no real effect, which was a little bit surprising. We thought that this might have some kind of effect, but again, this is in sort of an unoptimized scaffold. Colistin on its own had somewhat of an effect to prolong life, but on its own, it's not very great, and I'll tell you why in a minute. But in combination of devastatin and colistin, we could dramatically improve survival of the animals. And importantly, we could do this at a colistin dose, which was a quarter of the human equivalent dose. And colistin is one of these molecules that it was discovered a long time ago and then shelved because it's super toxic. But it's a really great antibiotic. And so it's kind of been described as one of these drugs of last resort. And so with increasing frequency of antibiotic resistance, sometimes it's the only pill left on the shelf that's going to work. Doctors don't want to really prescribe it very much because it causes renal failure and all kinds of other toxicities. And so it's really reserved for only a few select cases. So if you can find another small molecule partner to work with it, so you can dose spare and instead of treating with a high dose of colistin, treat with a low dose of colistin, that would be a good thing. And that's kind of what we saw here. And so this got us really excited, and we got it thinking more and more about this idea of colistin as a drug of last resort. What if we were to think of a salmonella infection that is in most need of new antibiotic drug discovery, where the drug of last resort would be problematic because of toxicity, what would that bacteria be? And this got us really quickly thinking about a type of salmonella called invasive non-typhoidal salmonella, or INTS for short. Now let me tell you what INTS is in case you haven't heard of them. So this is the collection of salmonella infections that we typically see globally. These are the papers that described all this work, so they're actually quite interesting. Globally, there's about 110 million salmonella infections a year. Obviously, this is underreported because people get salmonella infections and don't actually report it, but this is kind of what we're thinking. So the totality of salmonella infections would be about here. The vast majority of these salmonella infections are gastrointestinal infections associated with things like Salmonella typhimurium, Salmonella enteritidis. So these are non-typhoidal salmonella infections. Out of this 110 million, 95 million of them are these cases. So these are self-limiting. These are globally distributed strains which cause gastroenteritis. Salmonella typhimuriums everywhere in the world. About 14 million of these cases are caused by Salmonella typhi and Salmonella paratyphi, which are restricted only to humans. They have no animal reservoir. They're not zoonotically transmitted. They're human-human transmission only. And then there's another smaller group, so by comparison, a much fewer cases, but still 500,000 cases a year, of these diseases called INTS disease, caused by invasive non-typhoidal salmonella. So these are not typhoid or paratyphoid, but they're invasive, and so they cause invasive bloodstream infections like typhi, but they're not typhi. The vast majority of them are typhimurium and enteritidis strains. Why they're a problem is because they kill way more people than anything else. So gastroenteritis, salmonella which causes gastroenteritis, massive amount of cases. The case fatality rate is tiny, 0.05%. Typhi has a higher case fatality rate, but these INTS strains have a super high case fatality rate, 15 to 35%, depending on the age group. So overall, it's about 15% case fatality. In kids under 5, it's about 35% case fatality. The reason why they're so fatal is because they're bloodstream infections. They are associated with comorbidities like malnutrition and HIV infection. And they're associated with infections in kids under 5, which don't really have a super robust immune system. And they're multi-drug resistant. So there's not a lot of medicines available to treat them. Okay, so we thought, well, let's try and figure out a way to kill INTS. And so we've set up a whole series of experiments now in the lab to try and go after this. And this is some of the things I'm going to talk to you about in the last little bit that I have. So INTS disease is globally distributed. However, it's centralized in sub-Saharan Africa. That's pretty obvious in this graph. So this is an incidence chart. Sub-Saharan Africa has the highest incidence rates of INTS, but other countries report INTS disease. Even in Canada, we have INTS strains circulating. And so this is a problem because it's only a matter of time before this is even more globally distributed. And with more drug resistance, I do think that this is going to become a bigger issue. Again, incidence, this is globally. Incidence is growing. So these were some data from this paper here. In 1990, they were like 350,000. Now they're over 500,000, like in sort of around this time. Deaths go up. And the disability adjusted life years is gigantic with these strains. So years of life lost plus the years of life lived with disability Around now, it's like 4.7 million disability-adjusted life years annually. So it's huge, huge issue. I put this paper up just because I know a lot of people in the audience are interested in the One Health concept. And so this was a recent paper that was just published last month on INTS in two regions in sub-Saharan Africa from a genomic assessment of a One Health perspective. So they were looking at the transmission, and so where does INTS come from? They were questioning whether there's a potential animal reservoir, an environmental reservoir, is it strictly human to human? So if you're interested in the One Health and nasty bacteria, I think this is a really interesting paper that you might want to check out. I told you that the vast majority of disease, the burden is in kids under 5, and this is showing us this here. This is incidence per 100,000. Kids under 5 are here, here, and here. So there's a little bit of a blip also in sort of, when people are 35 to 50 as well in terms of incidence, but the vast majority happens here, and and this is where most of the death happens as well. Without a vaccine, or I should say, so without a vaccine, between now and 2038, there'll be over 9 million new cases of INTS disease worldwide. 3 million of these cases will be children who will die. So this from today until 2035, every single day, 330 people will die every single day, every day, until we solve this problem. So this is why there'd be a new vaccine or new therapeutics. New therapeutics won't prevent the infections, but they could certainly prevent the deaths. So that's what we're trying to go after. Antibiotic resistance is a massive problem here. So these are different types of INTS strains, so non-typhoidal salmonella strains. These are the antibiotics listed across here, and then it's a heat map of their resistance rates. So again, you probably can't see the numbers, but they're posting some pretty big numbers here. So there's lots of serotypes which have 100% resistance to, let's say, fluoroquinolones, which is a massive problem. Enteritidis is like 95% of them are resistant to fluoroquinolones. Lots of other big numbers here for antibiotic resistance rates in these strains, which is why it's really hard to treat them. You'll see here colistin, which we've talked about, almost no resistance per se to colistin. However, and so you could think, well, what's the problem? Just treat these people with colistin. Well, it's toxic for one, and there's a second problem. The second problem is, so this is work that we did, when you grow INTS in LB, it does have a pretty nice MIC against colistin. The MIC is like 1 microgram per mil. That's amazing. But the bacteria are not growing in LB when they're infecting someone under 5. When you grow them in these host mimicking media, which I told you about before, which we set up our screen with, the MIC shifts for colistin to between 32 and 64 micrograms per mil. That's impossible to treat in a human because renal toxicity starts at 2.5 micrograms per mil in the plasma. So you kill the person with colistin before you ever killed them, before you ever killed the salmonella. And so you've got this, they don't have necessarily acquired resistance to colistin, but they have this intrinsic resistance when they're growing in the host setting. And so that's a challenge. So how do you get over that problem? Well, you can do it with drug-drug combinations. So drug-drug combinations can overcome this problem of toxicity by dose sparing one of the drugs. And so I've showed you these checkerboards before. If you look at something if you have two molecules which are synergistic with one another, drug A and drug B on their own have very weak or no inhibitory concentration on their own. But when you start putting these two molecules in combination with one another at different concentrations, you get to a concentration of drug A and drug B, which is really good at inhibiting. So they work together, not alone. And so we thought, what if we could come up with a screen to dose spare colistin, so we didn't have to use so much of it in a human, so we could avoid the renal toxicity, and we could overcome this intrinsic resistance problem. What if we could find a small molecule that works in partnership with colistin that solves both of these problems at once? And so that's what Melissa Spiegel, who's a PhD student in the group, is working on at the moment. And so she's developed and implemented a workflow for drug screening for anti-INTS drug combos. And so I'll explain to what she's doing. She's taking a compound library, which we call the Bioactives 2. It has about 11,500 molecules in it. These are small molecules, some antibiotics, natural products, FDA-approved molecules, a whole bunch of other things. They're bioactive, they're known drugs. She takes the Bioactives 2 library and she screens it against an INTS strain all by itself to look for molecules in the Bioactives 2 library which kill Salmonella all by themselves. And then she screens the same library in the presence of a quarter of the MIC of colistin, so 8 micrograms per mil in these host-mimicking media conditions. So that concentration alone does not kill Salmonella, and so colistin by itself in this screen does nothing, but in combination with perhaps one or more of these 11,000 molecules, maybe we can find some combination that works great. So again, she does this in host-mimicking media, again, to activate these cryptic in vivo essential targets. We do the echoacoustic droplet dispensing, where we can dispense tiny droplets of small molecules into these wells. We do robotic handling for all the steps. And then we do, in this case, an optical readout at 600 nanometers. So she's done this work, and this is the primary data. Okay, so this is the Bioactives 2 library by itself. This cloud, so the X here is the index. So these are, each dot here is a single small molecule in the Bioactives 2 library. So there should be 11,500 dots in this plot. Most of the things do nothing. So this is the OD value. This is normalized optical density. Most of the molecules do nothing. The bacteria are growing just fine. So this is the OD, the cloud. Some things make it grow a little bit better, which is kind of weird, but we're most interested in things which kill. This gives us 194 hits. However, when she screens in the presence of sub-therapeutic doses of colistin, which don't do anything on its own, we increase our hit rate to almost 800 hits. So we get a vast majority of new small molecules which only work when colistin is present. So what are these things and how do we identify them? So first she identified what these single synergistic hits are. And so she did this by plotting the normalized growth with no colistin over the normalized growth in the presence of the combination with colistin. And so again, we're only interested in things down here which we've colored in green, which have only an inhibitory effect in the presence of colistin. So these are the guys in here, and gives us 635 hits. When you look at what these hits are, you can take the structure of these small molecules, you can arrange them in a linear sequence of characters which define the atoms and the bonds and the chirality of the molecule. We call them SMILE structures. And then you can feed those SMILE structures into a computer and ask the computer, how similar are all these structures to one another? When you do this from the Bioactives 2 screen alone, so no colistin, the vast majority of the molecules bin into a very limited set of chemistry, which are mostly known antibiotics. So big group are the cephalosporins here. We've got nucleoside analogs like AZT, which is known to inhibit growth, fluoroquinolones, for example. I would say expected chemical diversity. These things look like antibiotics because they work by themselves without any other partner antibiotic. And so that's what we found for this screen. However, when you do the same thing in the presence from the data with colistin, the chemical diversity of the hits you get becomes massive. And so there's way more chemical diversity. So the closer these dots are to one another, the closer they are in structure to one another. And so lots of interesting diversity here, lots of chemical diversity, lots of things for a medicinal chemist to get excited about when we show them to them. And so this is really, I think, really cool stuff. Okay, so how else did we then continue to prioritize? We prioritized molecules which had the greatest effect on inhibitory activity in host mimicking conditions. And so again, this is just the index plot of this subset here now. So we're whittling it down into a smaller and smaller set of molecules. And we're looking for things which inhibited here the most. And so we're looking for things which are not weak inhibitors, but good inhibitors of growth. So in this case, we set a cutoff of fourfold change in OD. That became these molecules here, which gave us 184. We then took all these 184 molecules and cherry picked them out of the compound collection and did dose response on them all. And so we did a series of dose response starting at 50 micrograms per mil, going down to 0.7 micrograms per mil. And so, for example, 80 of these compounds inhibited at 12.5 micrograms per mil. We were interested in molecules which had a better MIC than that, and so we focused our attention on these molecules down here, which in the presence of colistin had a really impressive MIC of sometimes less than 1 microgram per mil. And so that gave us these green guys here in 33 molecules. We focused on these 8 molecules and did, again, these checkerboard assays to look at what kind of synergy can we really see with these molecules when we have them in the presence of colistin. And so we picked 8 of those really top performers and did checkerboard assays on them here. So I'm just reminding you what this checkerboard looks like. Synergy is when you have a fractional inhibitory concentration of less than 0.5. And that's how you calculate fractional inhibitory concentration. Basically, it's an index number which indicates how good do these 2 molecules work together. And so if it's less than 0.5, that's what's called synergy in the drug discovery world. And so for this first compound, in the presence of, again, increasing concentration of colistin, increasing concentrations of compound 1, colistin, again, on its own, I told you before, has a pretty lousy MIC. In this case, 64 micrograms per mil. That's basically resistant. But then when you start dialing in this partner compound, you can reduce the MIC of colistin down to 1 microgram per mil in these host mimicking conditions. And then Melissa did this for all of the 8 compounds that we prioritized, and all of them show very, very impressive synergy with subtherapeutic concentrations of colistin. So characterized by this classic checkerboard staircase, ladder staircase in a checkerboard assay. FIC indexes well below 0.5, except for this one. This one actually did not verify. So we have 7 promising candidates now to show to a medicinal chemist and say, what can we tailor? What can we optimize? What can we change? How do we make this better? How do we make it more soluble? All the things that we would now need to do in a drug discovery campaign to move forward with these. We have not put these molecules into mice yet, which would be like something my lab would do. You know, we kill a lot of mice in my group, I think. So that's next steps. We're in the process now of gearing up for those experiments to see if these really work in the context of a real host setting. I'm a glass half full guy, so I'm hoping that this is all going to look good. So far, it's all looking very, very promising. So mouse studies will be forthcoming. In the meantime, though, what we have done is done something a little more simple, which again is asked, do they work when we infect Salmonella into a macrophage? So again, I told you before, Salmonella can get intracellular, and when it grows and replicates inside a vacuole. And so we set up an experiment where we seed macrophages, we infect them with Salmonella, we kill all of the extracellular Salmonella, which didn't get inside the cell, by applying an antibiotic. And so now the population of bacteria we're interested in is the intracellular population. And then we treat these infected macrophages with compounds. So in this case, it would be our compound that we've discovered in the screen in partnership with colistin at sub-inhibitory concentrations. And eventually, over time, we would let that cook for a while, lyse the cells, and count how many macrophages survived. And so when you do this, it's, again, quite impressive. And so control on its own, so untreated, you get a nice fold change in INTS Salmonella replication inside macrophages. Colistin by itself has a bit of an effect, but not a great effect because of the reasons I told you about before. It becomes intrinsically resistant when it gets inside a host. Compound 1 has more of a variable effect on its own in these kinds of experiments, but in combination with colistin has a very profound and striking and reproducible inhibitory effect on intracellular replication of INTS. And so we're really excited about this work. We're moving forward with it. We're actually thinking of expanding the original screen from beyond 11,000 molecules in the bioactives to a bigger data set, because we have a really scalable assay. So you could do this on 50,000, 100,000 molecules with relative ease. And so those are the kinds of things we're working on now. Okay, I'm going to summarize to say that small molecule screening under infection relevant conditions, that's what really we've been doing for the last 5 or 6 years. It all started by us thinking about how do we unlock cryptic gene targets? What are the gene targets which are involved in immune evasion? Those are in vivo essential. Maybe we could go after those. And we think that drug screening under these kinds of scenarios does hit bacterial targets which are cryptic in most modern drug discovery campaigns. I will say these in vivo drug targets, though, are still untested in therapeutic treatment. We don't have an antivirulence molecule, for example, that's sort of reached the clinic. Lots of things in clinical trials, but the in vivo drug targets, like, for example, antivirulence genes, are really still kind of untested. So, I mean, that's a caveat, but I think it's a really promising area. Antivirulence compounds can sensitize Salmonella to host defense immunity and are effective in preclinical mouse studies. So these were things which I showed you, if we treat with defastatin, for example, we can resensitize to antimicrobial peptides. I mean, that could be another really promising area of future investigation. And I've also shown you that drug-drug synergy, like our molecules with colistin, or drug-immune synergy, which I didn't talk about, but we've actually worked a lot on this, is how do some small molecules work in combination with the chemistry of the innate immune system to overcome acquired and intrinsic antibiotic resistance. And for now, our focus in this area is on invasive non-typhoidal Salmonella because it has such a global, it has such a dramatic incidence and disease and mortality rate. It's profound. It really needs some therapeutic intervention. So huge unmet need, in my opinion. Okay, this is my group at the moment. All these talented folks, some of which I told you about their work. So Melissa, I told you about that last half of the story. She's been doing the screening that I told you about, which was unpublished so far. A whole bunch of other people, which I told you about along the way. Collaborators, some of which are here, some are at McMaster, drug discovery, funding, and I'm happy to take questions for a while if you need.